We're going to talk about carbohydrates now and explain some of the basics of that and how that impacts sheep nutrition. So first of all, what are carbohydrates? Well, the building block of all carbohydrates are monosaccharides, which are single or mono sugars, saccharides. Examples of this would be glucose, fructose, galactose, xylose, ribose. The most common one that we'll be talking about is glucose. Monosaccharides can be bonded to either other monosaccharides in order to make disaccharides. Disaccharides, two sugars. Examples would be sucrose. So for example, the white sugar you put into your coffee or tea is a combination of a glucose and a fructose to make sucrose, or a glucose and a galactose together will make lactose, and that's a milk sugar. Then we also have polysaccharides. And polysaccharide, poly means many, again, sugars, and examples of these would be starch and cellulose. So what is the difference between starch and cellulose? Basically, if we stick a bunch of glucoses together in certain types of bonds, they make starch, and another type of bond, they become cellulose. For starch, it's called an alpha linkage. This alpha linkage is not super strong, so it can be broken relatively easy. The enzyme amylase, humans, we have that in our saliva. Ruminants don't have very much in their saliva, but some of the bacteria do have it, and also they in the small intestine have the amylase there so they can break down any starch that escapes ruminal degradation there. By the way, the bacteria that do that are called amylolytic because they break down that amylose. Cellulose, on the other hand, is what's called a beta linkage, much stronger bond, much more difficult to break, takes very particular enzyme in order to do that, and in actual fact, ruminants do not have this enzyme to do it. It's the bacteria or some of the microbes that live in the rumen, the fibrolytic bacteria or the cellulolytic bacteria in the rumen that have the ability to break that down. So the bacteria will break that down, and then they can use that as their energy source. So what are the carbohydrates for? The predominant use of carbohydrates for ruminants is going to be for energy. Anytime you break a carbon-carbon bond or a carbon-hydrogen bond in an organic compound, the energy is going to be released. We get to produce ATP, which then can fuel the animal's ability to walk, for its heart to beat, for it to be able to consume feed, for it to grow, everything that would require energy, producing milk, everything. Carbohydrates can also be used for building blocks for other compounds like amino acids. About 10 of the amino acids the sheep can make in its liver if it doesn't have enough of them, and so it can use sometimes certain carbohydrates to act as kind of a building block to make those amino acids. The rumen bacteria can actually produce all 20 amino acids, and they can produce those, again, as long as they have ammonia and those appropriate carbohydrates or other products that they need as those building blocks. The other thing that the carbohydrates are required for is to maintain gut health. And this is especially important for the ruminants. Remember that with ruminants, what actually causes them to ruminate is the bacteria scratching. I'm going to spend a minute talking about this for a minute. So it's important that you understand that those bacteria, when they're in the rumen, they're actually producing volatile fatty acids as their waste product. I produce carbon dioxide as my waste product. If I run and I can't breathe enough and I turn into anaerobic exercise, I will start to produce lactic acid in my muscles. That's the burning I feel. Bacteria, because it's an anaerobic environment in the rumen, are producing those volatile fatty acids right off the top: acetic acid, propionic acid, and butyric acid. And some will also produce lactic acid too. So you think about that rumen pH, it's going to be dropping and dropping and dropping as long as those bacteria are working. If it happens too much, the pH will drop low enough, like it does in your silo when you're making silage, and you'll kill off all the bacteria and the animal can no longer consume any more feed. And so how does that animal maintain that rumen pH? It does that by ruminating. In its saliva, it has bicarbonate. That bicarbonate gets mixed with the food that they're re-chewing, and then goes back down into the rumen and brings that rumen pH and maintains that rumen pH, buffers it to keep it where it's supposed to be. The other thing, of course, is those volatile fatty acids are also being absorbed through that rumen wall, so that some of the pH can rise again as those acids disappear from that rumen area. Now, what is it that actually tells a ruminant to ruminate? It's actually literally the physical scratching of the rumen. Right around where the esophagus comes into the rumen, there's an area there called the cardia, and as there's Physical scratching, it doesn't matter what it is. There's been research trials where they've used toothbrush bristles. Toothbrush bristles is enough. It's not the nutrient component, it's literally the physical scratching that causes that animal to ruminate the feed. Now you understand the importance of there being enough fiber in a diet and why we have to maintain enough forage in that diet to maintain rumen health. I really like this diagram. I think it's a great diagram, the way it shows the plant carbohydrates, and I like how it also shows the fractions and when we analyze them as well. So plant carbohydrates can go all the way from the cell contents all the way to the cell wall, and it's the other thing that is important. Organic acids technically are not carbohydrates there, but they can. That's included as being extremely soluble type of carbohydrate or energy source that's available for them. Then we have our mono or disaccharides. Generally, there's not going to be a great deal of that in a plant because the plant does make that through photosynthesis, but then they get converted into cellulose over time or into starches. And so the starches, again, it's going to be mostly in the grain portion of a forage, but there can be some also elsewhere. Then we get the fructans, we get the pectins, then we move into the cell wall. Again, that plant is trying to build cell wall as it grows in order to have the structure that it'll be able to stand in a field and continue to grow. As that plant matures, the products become more hemicellulose, then cellulose, and finally lignin. And lignin, we talked about lignified plants, that'd be a plant that's been really heavily matured. So when we analyze these, if we send these feeds off to get analyzed, if you ever hear the word ADF, that's acid detergent fiber, and we'll talk about that in a few minutes, it's referring to just that cellulose and that lignin on the far side, on the far right-hand side of that diagram. NDF would be your ADF plus your hemicelluloses. Then you have neutral detergent soluble fiber. The neutral detergent insoluble fiber is NDF, and the soluble fiber would be the stuff on the left. Fructans all the way to lignin would be considered fiber. Okay, or non-starch polysaccharides. And if we want to include the total starches, we would include also the total polysaccharides would include starches as well. And you can see the NFC there, that's your non-NDF carbohydrates that again are part of that plant. So this diagram kind of explains well, and as you can imagine, the stuff on the organic acid side, very digestible, very soluble in the rumen. Bacteria have very quick access to it, and then as we move further and further towards the lignin, it becomes less and less digestible. I really like this diagram too. This diagram shows quite well how energy has been used in ruminants, and so the energy can come in either forages. So I'm going to bring my marker up here, and you can see we have forages and grains coming in, and so we're going to have cellulose, hemicellulose, starches, and so forth. And I mean, there's going to be some glucose as well potentially, and those are going to then be broken down. If the rumen is able to break it down into the individual glucoses, then the bacteria will use those glucoses to produce, as I mentioned already, those volatile fatty acids. If that starch is not able to be broken down in the rumen for some reason, it can pass into the intestine where it can still be broken down into glucose and get absorbed through the intestinal wall as well. Any cellulose that escapes the rumen will essentially not be digested. Yes, there is some digestion that goes on in the cecum, but not very much relative to what the rumen is, and so it's going to likely just land up going through in the feces. And theoretically, starch can as well if it's not broken down enough into small enough particle size where it can be absorbed in that, broken down, and digested and absorbed in that intestine. Okay, so then we have these three volatile fatty acids there that are going to then go through the rumen wall. As they go through the rumen wall, they're absorbed through the papillae in the rumen wall. They move into the portal vein. All the blood that leaves the digestive system, okay, from the rumen right through to the large intestine, all that blood that leaves those tissues goes into the portal vein, which then goes directly to the liver. But anyways, as the butyrate goes through there, it actually gets converted into beta hydroxybutyrate, and so that is a ketone that you are probably aware of. Acetate, when it goes to the liver, it's used as an energy source. As a matter of fact, the predominant energy source for ruminants is actually acetate. That's what they will use. By the way, acetate also, acetic acid, that's what is in vinegar. Okay, so it's similar. It's the same acid. And then we also have propionate being produced, and that propionate actually in the liver gets converted into glucose. Think about it from a ruminant's perspective that's out there grazing on pastures. There's not very much glucose in there. If there is any glucose, quickly the bacteria are going to break it down. So how does the animal actually absorb glucose? There's going to be very little glucose unless they're eating a high grain diet where you have some energy escaping, some starch escaping that rumen. You're going to have very low levels of glucose available to that animal. Yet it still needs glucose for certain body functions. They absolutely require that glucose. And so what happens is propionate then go through a process called gluconeogenesis. Glucose, new, genesis, making. So it's making of new glucose out of that propionate. And then acetate and butyrate cannot go through that process. Only propionate can go through that. So then you get glucose made. The acetate can leave there. The acetate can go to the muscles and be used as an energy source in the muscles, or it can go towards be fat production. And if there's going to be some fat being laid down as well in the adipose tissue, you can see that either the ketones or the acetate. Glucose also is required for making fat because there's a glycerol backbone that happens there. And in the mammary gland, again, fat production happens through the ketones like the beta hydroxybutyrate, and also through acetate can go towards that milk fat production. And then glucose is used for lactose production. It's absolutely required for lactose production. Without glucose there would be no lactose. So you can see kind of how that all fits together, and how you can already start to see how we have to somehow optimize how much cellulose, hemicellulose, and starch is going to be digested in the rumen, and how much that starch is going to escape that rumen for digestion later. So let's talk a little bit. Lignin. Lignin is the stuff on the far right hand side there. It's completely indigestible. It actually comes from the Latin word for wood. Very indigestible. In most cases this is going to pass right through. So when you think about lignin, think about a tree. The bark on the tree, and the tree. I can grind that tree as fine as I want, and I can feed it to my animals, but they're not going to grow off of that. So indigestible. Even an alfalfa plant, as it matures, or any grass plant, any plant, corn silage, it becomes more and more lignified as it ages. The acid detergent fiber. So what this essentially is, just to make it simple, is what was developed was that we would use detergents, kind of not completely unlike a laundry detergent, again, a laundry detergent to wash the fiber. And so depending on how hard you washed it, you would get more or less fiber. Some of the fiber would actually become what we call soluble. And so an acid detergent fiber is actually where I add acid to that detergent. So it's an extremely strong detergent, and it will break down a lot of that fiber. So all we have left when we're done that is going to be the lignin and that cellulose, and that's all that's going to be left. As you can imagine, the more lignin and cellulose you have in it, that is going to be a good predictor of digestibility. So the higher levels of ADF generally means the feed is going to be lower in terms of its digestibility. So if I'm going out to buy hay, for example, and I get a hay analysis on it, I get an analysis done on that feed sample, and everything else being equal, I see a higher level of ADF, I just know that stuff is less digestible. Doesn't mean it's necessarily bad hay, because if it's, for example, for maintenance animals, then maybe that's what you want. It's also going to have more scratch then, because it's going to be more lignified, so it'll have more of that scratch that the animals need as well. Although NDF, as you see in a minute, will also take care of that. So neutral detergent fiber is essentially the same thing. Instead of it being acidic, it's a neutral detergent, not quite as strong, therefore more of it stays in that insoluble fat fraction. Of course, all the ADF is going to remain in there, but so is the hemicellulose. So essentially, if you take NDF minus ADF, you have a measure of the hemicellulose. Much more digestible cellulose. Now, all of this though, all that NDF, the hemicellulose, cellulose, and lignin, they're really going to dictate how quickly that feed gets broken down in that rumen and can move on. New feed cannot come in until the old feed leaves. And so this neutral detergent fiber actually is a good predictor of feed intake. And so we're going to have certain levels of NDF that an animal can handle. Once we go above those levels of NDF, then we start hampering feed intake on that animal. Tied to the neutral detergent fiber, we have something called effective NDF. Effective NDF is a measure of how well that NDF can actually cause the animal to ruminate. Not all NDF is equal. So for example, if I take something like soy hulls has a significant level of NDF in it. However, there's not very much scratch in it, so therefore you don't get that scratch and you don't get as much rumination out of it. Whereas something like, for example, straw has a very high NDF, and most of that NDF is effective at causing that scratch in that rumen. And so you'll also hear the term called physically effective NDF, PENDF. It's basically a measure of the ability of that NDF to cause scratch. It comes out of the dairy industry, where it was determined how much fiber was required in order to maintain a milk fat level of 4% butter fat. And the reason being that is that the higher fiber levels switches the balance of those volatile fatty acids towards acetic acid production, and acetic acid production will drive up milk fat production. So what are our sources of carbohydrates? Well, starches are going to be your corn, your oats, your barley, your wheat, your grains. Okay, the grains are going to be your sources of carbohydrates, or your starch carbohydrates. The cellulose is going to come from your grasses, your legumes, your corn silage. Okay, it's the whole plant, or the whole plant barley, or oatlage. Okay, those types of products, and also, of course, will also come from straw as well. So what types of factors influence that forage quality, whether we're going to be producing more ADF or more, a little bit more towards the NDF, a little bit more digestible feeds? There are three main factors. There's weather, there's the stages of growth, and then there's the species of the plant in the stand. So weather really has an impact in two different ways. It can actually affect the growth and composition of the feed, depending on how much water you have. And in droughty conditions, the plants will change their growth patterns. But also, perhaps even in a bigger way, it will impact your ability to harvest it at the correct stage of growth. So if I know, for example, that I need to harvest that hay right on such and such a day, but I know it's going to rain for the next 3 days after I get a cut, I might actually cut some of it beforehand. I might cut some of it early, harvest it early. It's going to be a little bit younger, which means the protein levels and the energy levels, the digestibility of it's going to be a little bit higher, but the yield's going to be a little lower. And then I may get stuck in the middle of that, and I have to harvest some stuff after the rain's gone through. Now it's become a little bit more mature, a little bit more lignified, and then therefore a little bit less digestible. So you can see how targeting the correct spot, we're always limited by what the weather that we get. There's often a very narrow window again to harvest that. And again, what the optimal maturity is, I can't tell you what the optimal maturity is because it depends what you're feeding. If you're feeding a lactating ewe, I want it to be a little bit more digestible and a little higher nutrient content. If it's for maintenance sheep, I want it to be a little bit more mature, and I'll get a higher volume off that acreage. So it really depends on what you do that. There are factors that can impact it. Again, we can't control the weather, but we can control, for example, the swaths. And I have wider swaths, but wider swaths isn't always necessarily the answer. When I'm cutting my hay, I have to pay attention to what the weather is. If it looks like, first of all, if my ground is dry and it looks like there's going to be a pretty sunny day, hot sunny day, I want that swath spread out as wide as possible. That way the sun can impact as much of that hay as possible and dry it as quickly as possible. If, however, it's going to be a little bit cloudy, but maybe some wind coming through, then maybe I want a narrower swath that's piled up a little higher. Now the wind can go through and dry it. If I have a pile of hay, then the bottom stuff is not going to be impacted by the sun, but if it's a little bit cloudy, then that's not going to be such a big deal. Then maybe I'm going to get more drying from the wind. If my ground is wet, then maybe I want to have a more narrow one as well, because again, the moisture is going to be coming out of the ground and hitting that forage from the bottom, so it's going to have a harder time drying as well. So in that sense, maybe a more narrow swath would be appropriate as well. So it's a little bit more complex than just saying one type of swathing is better than other. It depends on the conditions, your field conditions, and the weather conditions that you're dealing with at that time. Again, this diagram just kind of shows you that as that plant matures, NDF goes up and ADF goes up. And then there's different numbers that are used, measures that are used. This one's, I believe, the relative forage value, okay, or the relative feed value of it. And you can see as that plant matures, the relative feed value drops. The protein levels drop, the energy levels. It's not necessarily that the gross energy drops. We'll talk about gross energy versus digestible energy. The total energy in there may be the same. However, the ability of the animal to use that energy will decrease. So that's why it's a relative forage value. Now, other factors that impact it is going to be, for example, legumes versus grasses. Young grasses have a very high feed value, but they change more quickly than legumes do as they mature. So grasses start very digestible and high energy, but then as they mature, their feed value of those will drop. Okay, the ADF and NDF climb relatively quickly, whereas alfalfa a little bit lower than a young grass, but it maintains itself, so it doesn't take very long then when it gets a little bit older or in mid-maturity that it surpasses the grass in terms of quality. The other factor that can affect it is species within each class. So for example, timothy versus orchard grass versus bird's foot trefoil versus alfalfa, clover, and so forth. And there's an excellent document that we have available to us here in Ontario. It's called the Agronomy Guide for Field Crops. I highly recommend that you have that as a resource. Now you can download it online, or you can purchase a book, a copy of it, and it has an excellent description of the many different species of forages and some of the strengths and weaknesses of including each of those in your stand.